Jürgen?
Hey, Nora.
Hey.
Hello.
Hi.
Let me see.
How's everybody? Good?
So far, so good.
Awesome.
Me project out. Okay, there we go.
Okay, perfect. What's new in Washington?
I have no idea. And that's a good thing that I don't pay attention.
That's good. That's always good.
Yeah. No, lately I've just had to deal with small kids at the daycare and just.
Yeah, that seems like a lot of work. Hi, Sarah.
Hi, how are you?
Good. How are you?
Good.
Okay, we got Aseda.
Are you still at work?
Yeah, I just
Always,
finished up.
always.
Sarah is always at work.
Yes.
3 more weeks.
Yeah,
This
like
place.
9:30 p.m. No, she's there.
Hi, Aseda.
Hello, guys. Good
Hi.
evening.
Good evening. How are you?
I'm good. How are you?
I'm good, thank you. You're at work too.
Of course. Can't leave the lab.
Okay.
Hi, Aseda. How are you?
Dr. Sawushai, how are you?
Excellent.
You look great. Thank you for coming.
I wouldn't miss it.
I feel so honored.
It's
Looking
past my bedtime, but I stayed up.
We'll move it, Dr. Chawushai, earlier.
No, I'm
You
teasing.
want it earlier?
I want you to be in every journal club, really.
I think it's past Erion's bedtime. That's who I think.
Oh, yeah, that could happen, yes.
Is that why he's not on yet?
You know, you've done such a wonderful job. I didn't know there were that many Albanians. I'm so happy. I knew Aseda. She's my pal.
Yeah.
Yeah.
Well, I'm
Yeah,
amazed
beautiful.
too, because people are just approaching and they want to present, and I'm like, yes, we want more. Bring us more. It's fine. We will help organize it. And thank you to everyone. Thank you to Sarah. She's amazing. She really makes it happen.
Oh, yeah, that's really
We have such a great team here. It's
I bet you do. There's
Yeah.
a doctor. Her name is Mylinda Madden. Although she's from Albania, she just started at my hospital, and she's wonderful. How do we reach her to join?
Is she a resident?
No, she's an attending hospitalist.
Oh, really?
Yeah, she's fantastic. I love her. Name is Mylinda Madden.
Can you
I've
spell
Googled
her name?
her, but if you have her email and want to send it,
I will torture her this
yeah.
week.
Wait, let me see. I think somebody approached me actually through LinkedIn. Maybe it's her. We're supposed to talk. My Linda, huh?
Yeah, Mylinda Madden. She's a hospitalist.
Oh, okay. No.
Madden is her ex-husband's name. I told her not to change it back to the Albanian.
What?
I'm joking.
Okay.
Joking.
It's probably the American one is probably easier to pronounce, so it's okay.
I've never changed my name, and people call me cow shit. So it's
It's okay. I've got crazy, so
Have you ever corrected them?
is that Dr. Crezio?
yeah.
Excellent. How are you? How's
Good. How are
your
you?
residency doing?
Good. I'm excited to start up again in July.
I bet you are. You're on the gastric tumor service at Memorial.
No. I mean, in the lab I am.
In the lab. Oh, okay.
Yeah, in the lab I'm in the sarcoma lab, but
Yeah.
I'll be back at NYU in July.
Have you met Dr. Aguilar?
I have not. No.
Well, I'm having dinner with him in 10 days.
Oh, lovely.
So I'm looking forward to that.
Yeah.
You probably know Dr. Ramsey and Dr. Bernstein.
I'll be with them for 2 months next academic year.
Don't tell Dr. Bernstein you like me, because I trained him.
He just stepped down as like chief of the colorectal service at NYU.
Who took it over?
Nobody yet, but I'm guessing Dr. Ramsey.
That's what I'm guessing too.
Yeah.
The Turk killed the Jew. That's the way it works. I don't know.
I don't know either.
I'm being recorded now, so it's okay.
Yeah, we'll cut this part out.
No, that's okay. That's okay. That's okay. Dr. Ramsey calls me out on out, so I don't mind insulting.
He calls me that too.
Yeah.
Wow.
You know why? It's a term of derision.
No, I was hoping that wasn't the reason.
That is the reason. But you know what? You're in proud company. Don't worry about it.
Hi, Dr. Chaya.
Hello there. How's everyone doing?
Erion, I stayed up past my bedtime for you.
I know you did.
How you doing, buddy?
So did I. Good. How are you?
We miss you at Hartford Hospital.
I miss you guys a lot. Tell hi to the boss.
I will say, I actually saw him today. I insulted him a little bit, which is my way.
That's so unlike you.
Yes, but I told him I was going to see you tonight, and he didn't believe me.
Yeah. Well, yeah.
How are things going there for you?
Pretty good. Can't
It's
complain.
a good hospital. It's a good.
Yeah.
You're in a good system. You're in a good place.
It's a smaller pond for me. Something I was looking for.
Yeah, no, that's right.
Yeah.
So you'll be interested, since your father was the dean at Chasut. I'm going there this summer, but starting September, I will be having my own clinic at the medical school there and seeing patients with coloproctological problems. And
That's
teach-
nice. Yeah.
teaching, and they asked me how much I wanted. I just said I need a room and occasional coffee.
I doubt that's it, Dr. Chaushai.
Well, I'll have a beer every now and then too, but you know, so I'm looking forward to it, actually, to do something good for the poor people of Albania.
It's awesome.
I know you mean well.
Thank you. My son has instructions if I die there just to burn me and bring my ashes back.
Don't say that.
Why do you have to get dark at the beginning of a journal club like that?
Aren't all journal clubs dark and, you know?
After all, we all
Did
talk
you
about
know,
COVID.
did you know your successor, Caesar?
I know of him, yeah.
He has no sense of humor and he's never seen Planet of the Apes. He thinks I was insulting him by saying that he reminded me of someone from Planet of the Apes.
I hear more stories that I like to hear from that place, so.
Yeah, I'm not crazy about Caesar. I knew his boss. His boss was a resident of mine at UMass.
Yeah.
Who we cut appropriately so, back in the good old days when men were men and sheep were afraid to come out at night. Wow. Is there anybody still listening? Because I can scare away everyone. Nora, are you still there? Am I alone?
No, we were like about 16 now.
16, excellent.
Yeah. I'm sure they will join us more.
And I'm sure before we start, sorry to interrupt, Dr. Chawusha, but
Yes,
I really think these journal clubs have to be as interactive as they can. So on that side panel, if you have a question, you're hesitant to interrupt, just write the question. We'll make sure you get answered at the end. There's no stupid questions. There's really stupid answers which fall upon me. So go ahead and fire away.
Bravo. You are absolutely right.
That's great.
Before are we about to start? Can I just compliment our president?
We have time for a joke.
No, this is not a joke. I mean, having been the fourth and the lousiest president of the society, what Nora has done in 6 months has been revolutionary. The journal clubs have revolutionized the society, the membership, and the baton appropriately has been passed to a new generation. And I'm so very proud of our current president.
I second that thought.
Thank you, Dr. Chaushai.
We love you, Nora. You're fantastic.
I have a great team. Everybody's been so helpful, and this is how we're thriving all together.
Are you in Hawaii still?
I am, yes.
Excellent. That's even better reason to like you. I love you. You're out there, you're hanging out with Don Ho, and you're having a good time. Good.
Yeah, it's my last month though. I'm coming back July, so I'll be in
Then
East
where?
very soon.
Where will you be in July?
I'm coming back to New Jersey. That's where I have my condo.
Excellent. Good for you. Good for you.
Yeah.
Where in New Jersey? I used to live in Fort Lee until Woody Allen said that God did not exist there.
Okay.
Are those news?
So I think I timed that interaction for everyone.
Did I offend someone?
Sarah, that slipped away from you.
Say that again.
That introduction slipped away. I don't know. We get in a second conversation in the background.
So yeah, I think we have enough people. I'll let Dr. Gashi get started.
Yeah.
Okay, everybody's good?
Yes, ma'am. I'll be quiet.
Is Aseda here? Because I don't have any slides. I'm sorry.
Yeah, she's here. She's here.
Yeah, Aseda's here. She's here.
Okay, well, good evening, everyone. Welcome and thank you for joining AAMS Journal Club tonight. I am Nora Gashi, President of Albanian American Medical Society. I'm very excited to see so many Albanians, healthcare professionals interested in participating in these events. And I encourage everyone, please, if you have any new ideas for Journal Club, send us suggestions and we'll be more than happy to organize it. So tonight's topic will be about AstraZeneca COVID-19 vaccine and one of the reported adverse events. When Aseda approached me about the article and asked me if she can present it, I thought it was an excellent idea. This was a very good article, and I think we need to discuss the possible side effects of various vaccines. So we need to be able to explain to our patients the risks and benefits of vaccines versus the risks of disease itself. So tonight's article, which was recently published April 16th in New England Journal of Medicine, Pathologic Antibodies to Platelet Factor 4 after ChAdOx1 nCoV Vaccination. Presenter, Aseda Tena. She's a PhD student in biological and biomedical sciences and leader human biology and translational medicine program at Harvard Medical School. She has a long-standing scientific training in basic science and translational medicine, encompassing the fields of immunology, transplantation biology, genetics, and cellular and molecular biology. And Dr. Erion Qaja, he's a vascular surgeon attending who will act as the discussant for the presentation and share his expertise and experience. Dr. Qaja is a vascular surgeon who finished vascular surgery training at Hartford Hospital, University of Connecticut, and currently working at Nuvance, Health Division of Vascular Surgery in Poughkeepsie, New York, since August 2020. Okay, Aseda, it's your turn.
Right, we're on.
Yeah.
Am I able to share the screen?
Yes.
I still have disabled coming up.
So if you try right now, it should work.
Yes. All right. Does everyone see this?
I do, yeah.
Yes, okay. So thank you, Nora, for the kind introduction. And I also want to give special thanks to Jürgen and Sarah for organizing this Journal Club today, and also Dr. Erion Qaja for being available to answer the clinical questions as they come along during this presentation. So in fact, Nora mentioned one of the papers that was published, but actually I found that there's two of them. And there was one from the German group that was published earlier, April 9th, and then the British group was the second one that followed these findings that are both related to the thrombotic thrombocytopenia events that occur in patients who receive the AstraZeneca vaccine. So this is really a very relevant situation right now because we need the vaccines to curb the COVID-19 infection, but in the same time, when these adverse events that are happening with the adenovirus associated vaccination, we need to address them properly so that patients cannot die from receiving these vaccines as secondary complications related to the vaccines themselves. So the important finding in both these papers is that they see a similarity between the HIT, or what is now the heparin-induced thrombocytopenia events that happens clinically when patients receive heparin. So it is very striking here to see that these patients in the serum, they have circulating antibodies, autoantibodies to platelet factor 4 that are occurring now in the absence of heparin. So this is called the autoimmune HIT, or autoimmune heparin-induced thrombosis. So we will discuss why this is happening in the end, but before. My slides got stuck. Here we go. So before we dive into the paper, I want to provide a review of what hemostasis is and how heparin has an anticoagulation effect when administered IM or IV, and how does heparin induce thrombosis or HIT. And then we'll talk about the adenoviral COVID-19 vaccines to see what is it about this vaccine that might trigger the autoimmune HIT in people who are receiving AstraZeneca vaccines. And then we'll talk about the results of the paper and then discuss furthermore the findings. So first, a reminder for everyone, hemostasis, or the process of stopping the bleeding. That happens when there is an injury to the vascular system. So upon injury to the vasculature, there are factors that are released from the endothelial cells that line up the vasculature that trigger a series of steps to create a plug or a clot so that the bleeding does not occur. One of them is the initial part is known as the vasoconstriction. Then there is platelet activation that create the platelet plug. This is known as the primary hemostasis. And then there's the activation of the coagulation cascade that requires the activation of thrombin that produces thrombin to produce a blood clot. In the same time, the coagulation cascade is also controlled by the antithrombotic control mechanisms because we do not want hypercoagulation to occur. So, we have coagulation happening, and then there is the fibrinolytic pathway also activated so that these fibrin clots or mesh are then dissociated at the end after the injury or after the repair has done to the injury site in the vasculature. So if we look at this more specifically, like we're taking all of the 3 different steps that happens during hemostasis, we can see that at the time of the endothelial disruption at the surface, when the disruption or the injury has occurred, the platelets, they bind to the endothelial cells by using the von Willebrand factor and the glycoprotein 1b receptor. Upon activation, upon binding, the platelets are activated, so they express these glycoprotein 2b and 3a receptors that bind to fibrinogen. So this in itself causes more platelets to get activated. And so a serial activation of a lot of platelets gets them to accumulate and stick together, creating what is known as the platelet plug. Now, platelets also release other factors known as tissue factors that can upregulate or activate the coagulation pathway. Now, there are other cells that are releasing tissue factors that also play a role in upregulation of the coagulation pathway. So there's a lot of factors here that are involved that are not necessarily to go through all of them, but the important ones are the factor 10 and factor 10A that are required to activate the prothrombin into thrombin, which then activates the fibrinogen into fibrin, and is the fibrin that creates these cross-linked fibrin clots that together with the platelet, create this plug at the site so that blood doesn't come out, and also other exposure, other ingredients do not go into the bloodstream. So both, as I mentioned before, the coagulation and the fibrinolysis cascades are very important to be well controlled because we do not want hypercoagulation to occur. Now, in the fibrinolytic cascade, the important players here are the plasminogen, which activates plasmin, and that directly has an effect into breaking down the fibrin clots themselves into known fibrin degradation products, or FDPs. Now we can measure these FDPs clinically through an assay that is known as the D-dimer. So this is important because the British paper really emphasizes that the initial screening for patients that are developing vaccine-induced thrombosis, thrombocytopenia, should be screened by the D-dimer. If they have really high D-dimer levels, then they are really they really have occurrence of VITT. So how does heparin come into play here? So heparin is a naturally occurring glycosaminoglycan and is widely used clinically for its anticoagulation effects. Now, more specifically, treatment for heparin are seen in DVT, PE, or cardiac surgery, and more. Now, heparin is very specific because it is the only glycosaminoglycan that is stored in the secretory granules of mast cells. Most of the heparin-like molecules, like heparin sulfate, is also stored in other cells in the body. Endothelial cells are also storing the heparin sulfate. And heparin only gets released at the vasculature site of injury. Now, heparin is synthesized as a long polymer with different molecular weights. Now, there is the unfractionated part of heparin and the fractionated part. The difference between the two is the length of the sulfate groups. So glycosaminoglycans, as a base, they have pentasaccharides. And attached to them are the sulfate groups. And how many sulfate groups you have attached to the pentasaccharide determines whether you are unfractionated or fractionated. So the longer sulfate groups you have attached to the pentasaccharides, the more molecular weight you have, and you are characterized as an unfractionated heparin versus fractionated with lower molecular weight and fewer sulfate groups. Now, heparin is known to cause the heparin-induced thrombosis, and the incidence of HIT occurrence are higher when patients receive the unfractionated heparin versus the fractionated form. Now, heparin itself, looking at the mechanism of action here, does not really break down the fibrin clots. Like tissue plasminogen, that in itself directly interacts with fibrin and breaks it down, but heparin doesn't have that effect. What heparin does is the activation of the antithrombin 3 enzyme that in itself inactivates prothrombin to become thrombin and fibrinogen to become fibrin. So the absence of both thrombin and fibrin activation leads to no clot formation. So that's why heparin is utilized as an anticoagulant. So looking at the pathophysiology of the heparin-induced thrombosis and thrombocytopenia, so why does this occur? So platelet factor 4, referring as PF4, is a chemokine that is produced by the alpha granules in the platelets when they're activated. So when heparin is injected, the platelet factor 4 binds to heparin and forms this complex known as the heparin PF4 complex that has the ability to be recognized by autoantibodies, more specifically IgG antibodies. Together, this antibody binding to the heparin PF4 complex creates an immune complex. And this in itself binds to the Fc receptors on platelets, which causes activation of platelets. So this immune complex causes platelet activation, and platelet activation through all the pathways we just said causes thrombosis. On the same time, this immune complex, when it coats the platelets, it also makes them prone to apoptosis, and the platelets are cleared by splenic macrophages, causing thrombocytopenia. Other than platelets, there are other cells that are activated by these antibody immune complexes, such as endothelial cells, monocytes, and neutrophils, which in itself exacerbates the HIT symptoms. But HIT is also an autoimmune disorder. And by that, I mean that there is an activation of these autoantibodies even in the absence of heparin. So when you are injecting heparin exogenously at high concentration, heparin now acts as a hapten. So that creates an autoimmune reaction to the circulating autoantibodies that we might have. But also we see these polyanion antibodies that are active to platelets, so they can activate platelets even in the absence of heparin. So in the paper, that's papers that we're going to discuss, all the patients that were affected by the vaccine developing these HIT symptoms, they never saw heparin. It was only one patient that received heparin, but all the other patients had never seen heparin, yet they had these autoantibodies circulating that could bind to platelet factor 4. So there's been a lot of theories of why can this occur? How come that we have these circulating autoantibodies? Or like even heparin, if it's an endogenously occurring molecule, why are we creating autoantibodies against it? Or like, why are some people more prone to creating autoantibodies and others aren't? So most of the papers have looked at the role of the bacterial or viral invasion because platelet factor 4 can also serve as a danger signal for the presence of pathogens. So what has been proposed is that platelet factor 4 biochemically is a positively charged molecule. So that makes it a cation. But bacteria, viruses, they have on their surfaces molecules like lipopolysaccharides, LPS, which is negatively charged. It's a very large molecule, is negatively charged, so it's an anion. So when bacteria invades us, we don't have anions in our body. So that in itself is a red flag that we have an invader in our body. So platelet factor 4 can bind to this bacteria, and this complex can be recognized by the B cells, which then creates antibodies to clear the affected cells. But then these antibodies can go on circulation. Now, heparin, on the other hand, why does it cause this autoimmune-like disorder? Because heparin is also another molecule that is negatively charged in our body. But what I think is happening here is that even though this is a negative charged molecule that is endogenously occurring in our body, we're giving it at very high concentrations that it's creating these really polyanions together that when bound to platelet factor 4, they are recognized by the B cells as the red flag signal, so the danger signal. So that's why the B cells are being auto-activated. So in the absence of the heparin, what causes is circulating autoantibodies is called the spontaneous HIT disorder. And this is when heparin is given to these patients and they receive and they make these antibodies, and this is known as the conventional HIT because heparin was administered. So heparin served as the heptan. So laboratories have developed a lot of assays that can identify HIT clinically, and also many algorithms. Sorry?
Sorry, I think somebody unmuted accidentally.
Okay, any questions so far, by the way?
Nothing so far. I'm checking that little chat. No questions.
Okay.
But please, feel free to write them down if you're hesitant to interrupt.
I mean, yeah, they can also interrupt and ask the question if something is not clear.
Well, you're doing fine. It sounds
And
fantastic.
either I'm explaining really well, or no one is understanding it.
No, you're explaining really well.
You're explaining very well. Thank you so much.
It's
Keep going.
fantastic.
All right. So laboratories have developed these diagnostic tests for measuring HIT, and devise strategy about how to manage HIT when that occurs. So the first assays I know are known as the immunoassay tests because they can usually run by ELISAs. They can measure these autoantibody complexes with incubation with platelet factor 4. And the other assay is known as the functional HIT assay, which is important to show that these antibodies have the ability to activate platelets in vitro. And for that, they use the serotonin release assay, SRA, because serotonin is also released upon platelet activation, or they can use the flow cytometry to look for activation markers. And this comes in handy because the British group used the flow cytometry to show the activation from these autoantibodies. Now, recommendations for patients that have been diagnosed with HIT is immediate discontinuation of heparin and initiation of a non-heparin anticoagulant at therapeutic intensity using non-heparin anticoagulants. And you need to discontinue platelet transfusion because by giving more platelet transfusion, you are exacerbating the problem and you're causing more thrombocytopenia. So now that we covered HIT and what HIT is, what is it about adenoviruses, COVID-19 vaccines that might trigger HIT events in people who are receiving these vaccines? So for everyone's edification, adenoviruses, they are double-stranded DNA viruses that cause respiratory or GI tract infections. And they've been widely used by molecular biologists for gene therapy. So most of the adenoviruses in the laboratories, they have been modified. By that I mean a certain gene is removed from the surface of the virus so that it doesn't replicate. So that this modification makes the adenovirus replicative defective. But it can infect the cell because it can still get inside the cell through these other receptors that it has on the surface. And scientists use this as a vector to deliver their gene of interest. In our case, it would be the spike protein, so that we can make antibodies against it for SARS-CoV-2 protection. Now, there are adenoviruses can infect humans. There are about 50 serological tests that have identified different 50 adenoviruses that can infect humans. And there are also adenoviruses that infect non-human primates. And what AstraZeneca has used is a chimpanzee adenovirus that has been, again, modified so that it doesn't replicate. And CH stands for, the species, chimpanzee. AD is adenovirus. ADO is adenovirus, and X1 is the number of the adenovirus that they have taken from infected chimpanzees. Within the, so the spike protein is inside the genome or the vector of the adenovirus, and this is released after the lysosomal degradation of the virus in the cell, which then gets incorporated into the cellular machinery to make messenger RNA, and then gets translated into protein. And these proteins are the ones that can elicit the neutralizing antibodies for COVID-19 protection. So, but what else could be inside these adeno vaccines anyway? So I have compared here side by side both the AstraZeneca and the J&J vaccines. Like, we're looking at the other ingredients other than the adenoviral vector itself. So AstraZeneca has the chimpanzee adenovirus, and the J&J has the adenovirus, the human-derived adenovirus known as the AD26. They both have this monohydrate salt, although this is more an amino acid monohydrate in the AstraZeneca vaccine. There is alcohol and surfactants because they are important to create this stickiness when the vaccine is induced, so that the adenovirus can clump together and survive longer. In addition to these commonalities, there are differences between AstraZeneca, like sugar and other salts that are present in here, but not present in the J&J vaccine. But none of these ingredients that are listed here are known to trigger an immunogenic response. They're not strong enough to trigger immunogenic response. So the only culprit here seems to be the adenovirus vector itself. But still, it is a mystery. How can the adenovirus activate platelets? The AstraZeneca vaccine is given in 2 doses. They are usually 8 to 12 weeks apart, and the J&J is only a one-time dose administration. Now, United States only uses the J&J vaccine, not the AstraZeneca, but even us have seen cases of VITT in people who have been immunized with the J&J vaccine. So back in April, the FDA and CDC put a hold on the vaccination because of the concerns of these severe VITT. But after reviewing all the data, they decided to bring it back into market, so people are still receiving that. Although again, they're closely being monitored for symptoms of VITT. So this is a summary recently from the BBC that shows the cases of VITT that the UK has seen. So around 35 million doses of the AstraZeneca vaccine have been administered in the UK up to May 19, 2021. And around 332 cases have been reported with these rare serious blood clots combined with low platelets. All of these symptoms were seen almost all after the first shot. Now, the incidence of having VITT is really high after the first shot, but that drops significantly after the second dose. Now, to put all of these findings in context here, there's only been 58 deaths from VITT in around 2 per 1.2 million doses, but there have been close to 2,000 deaths per million of COVID-19 in the UK. So the bottom line is you are more likely to die from COVID-19 rather than the complications from VITT after you're receiving the AstraZeneca vaccine. So it's still very important, underlying importance of receiving the vaccine because the COVID infection is still more deadly than the VITT complications. So now that we've covered all the background, we can dive into the papers, both of them, and discuss their findings. So before I do so, any questions so far?
No, brilliant review.
Great. So the first paper that I'm going to discuss is the findings from the British group.
Aseda, can you
Yes.
can I interrupt? I have a question here in the chat. Is it okay?
Sure, please.
Is it possible that the adenovirus interacts with one of the ingredients to give rise to these antibodies?
Not that there I know of any reports of. The German group does speculate that the adenoviral particles themselves can activate platelets because they've been shown in vitro or in mice models that the adenovirus can bind to platelets and activate the platelets. However, they're still under review. There is no direct link here, but I don't think there are the other ingredients that are causing these autoantibodies.
Just to clarify that question, Franz, what do you mean by the one of the ingredients? Is it the plasma cells? Is it
I think the vaccine components, what's inside the vaccine.
I see. Okay.
So any of those that were listed before is not shown or has been shown to be immunogenic at all. So, by process of elimination, the culprit here seems to be the adenoviral vector. But again, we don't know why the adenovirus vector itself can cause VITT because adenoviruses have also been used for gene therapy. Now, is it the concentration difference that the particle forming units that are in these vaccines are different from what is being used for gene therapy? Because in gene therapy, I think there's been only one case of reported complications with thrombocytopenia and thrombotic events. So clearly we're seeing these only with COVID-19 vaccination. So it's still really unknown why this is occurring. Like, why are these vectors selectively activating platelets and causing these autoantibodies against the platelet factor 4. So now coming back to the clinical summary or the laboratory findings of the 23 patients that were studied in the UK. So the median age of the affected patients ranged was 46 with a range from 21 to 77 years old. Now, 14 out of 23 were females, so 61% were females. All reported patients were previously healthy and fit. Now, symptoms developed as soon as 6 to 24 days with a mean of 12 days post-vaccination. What's worth seeing here is that the platelet counts is really, really low across all the 23 patients, so they're severely thrombocytopenic. Now, what the British group is emphasizing is that the fibrinogen levels are very low, but the D-dimer levels are extremely high in these patients. So because they are seeing this severe thrombosis with severe thrombocytopenia, that's why they realize that these analogy or these findings, these symptoms that are being presented are very similar to the HIT. So they did the ELISA assay to confirm that these patients from the serum that they obtained have circulatory antibodies that are binding to PF4. So similar to the platelet-induced HIT. Heparin-induced HIT, but in this case, none of the patients have had heparin before, yet they have these circulating autoantibodies against the platelet factor 4, which then activating the platelets together as a complex from these autoantibody complexes. they did this assay, like further corroborated the finding that this patient had indeed HIT-like symptoms by doing the functional HIT assay. So the functional HIT, again, measures the platelet activation, the ability of the serum. So the serum contains these circulating autoantibodies and the ability of these autoantibodies to activate platelets. So for those who are not familiar with flow cytometry and how this assay is set up, I wanted to give a short schematic here and then explain the gating here. So platelets from a donor with a group O are received, and they are distributed equally in all these different tubes, fax tubes. In the first tube, they would add the calcium ionophore. This is a known positive control because it's known to activate platelets. In another tube, they would just add platelets plus heparin. A third tube would have the platelets plus the serum, whether this is serum from control patient or the study patient. The other tube would have a serum plus heparin. So this will confirm that the circulating antibodies in the serum will be activated in the presence of heparin. So they are directed against heparin. And the last one is serum plus heparin excess. This is a control because if you give supraphysiological doses in vitro, you can modulate the platelet activation. But you cannot do this in vivo. You cannot give patients supraphysiological doses of heparin. That's unacceptable. But in vivo, you can do that. In vitro, you can do that and modulate and see that there is a modulation in platelet activation. So after the addition of each platelets and incubation with the corresponding activant here, the cells are then stained with CD41 and the Nexin-5 antibodies. Now, each of these antibodies has a fluorophore bound to it so that we can visualize it when we acquire these cells with flow cytometer. The CD41 is a platelet activation marker, and the Nexin-5 is a marker for apoptosis. So there has been a lot of studies showing that these autoantibodies can also cause apoptosis. So that was the clearance of these platelets by the splenic macrophages, the reticulocyte system. So let's look at the data now. So the control patient. So these two plots here are the positive and the negative control. Again, we're looking for CD41 versus a Nexin-5. So when the platelets are incubated with calcium ionophore, they are double positive for both markers. When they are incubated with only heparin, there is no activity here because we're not expecting to see activity in these platelets just being activated by heparin. Now, the serum for this control patient came from a patient that had confirmed conventional HIT. So what they did next over here is to incubate platelets with the patient's serum. So when there is the platelets plus the patient's serum that had HIT before, we still don't see platelet activation because you need also the addition of heparin to confirm that this is heparin-induced platelet activity HIT. So that's indeed what you see here. So when you add heparin to that sample, then you see around 41% of the platelets are activated in the presence of heparin. So this is autoantibody specific to heparin. But this is what happens in the study patient. You incubate platelets with the serum from these affected patients, you see 55% activation. So this is no serum, there is no heparin in here, just the circulating antibodies that are in the serum binding to the platelets activates the platelets. And when you add heparin, you see around 36% activity. So there's some cross-reactivity here with heparin, but exclusively seems like these, most of these auto antibodies that are circulating are independent of heparin. So something else has activated these autoantibodies that are activating platelets. So coming back now to the clinical findings of the German group. Now, this group had only 11 patients in their studies, and similar to the British group, they were pretty young. In their median age was 36, with a range from 22 to 49. Very strikingly, similar to the British group, 9 out of 11, 81% were women. So it seems like there is a higher propensity of women to develop these VIDT symptoms. And symptoms were developed 15 to 13 days post-vaccination. Now, in contrast to the British group, that they emphasize a lot on the high D-dimer level, the German group, when they do their clinical measurements of the D-dimer level in these affected patients, they don't see a very high level of D-dimer. They do see some increase in the fibrinogen, but not D-dimer like the British group did. But they do exclusively see across all of these patients these circulating PF4, heparin ELISA autoantibodies. So similar to the ELISA findings that were reported in the British group. So similarly to the British group, they also do these platelet activation assays. So when they do it in 4 patients, they don't see a lot of platelet activation in the presence of the platelet factor 4. So this is the key here, the buffer, which is the control, and then the platelet factor 4. The low molecular weight is the heparin, so you don't really see any activity against the heparin itself. But when you have platelet factor 4 added to the platelets, then you see activation. So these autoantibodies, again, are directly against the platelet factor 4. But when they compare this platelet activation assay, or when they did it across 24 patients that had these clinical VIDT symptoms, then they clearly see an increase in the platelet activity. And this is a little bit strange, or a new finding, at least for the German group, seeing that they see a lot of platelet activation when there is platelet 4 and heparin, almost comparable to the platelet factor 4 alone. But if you remember, from what I just showed on the FACS plots in the British group, it seems that the British group, or the findings there, the activity of the platelets was driven more, was higher when the heparin was absent. But here, seems like both the combination of platelet factor 4 and heparin activates platelets, similarly to just platelet factor 4. So these autoantibodies against circulating are clearly recognizing platelet factor 4 and cross-reactivity again with heparin. So based on these findings, they both have devised different algorithms about how to identify the patients that are affected and how to actually treat them. So the group here, this on the left side, this logarithm is from the German group. So what they suggest is that if the patient has thrombocytopenia and thrombosis between 5 to 20 days after vaccination, you do an immunoassay screening. So you do an ELISA screening. And if that is positive, you move further by doing the platelet activation, the functional assay. If that is positive, then then confirms the presence of VITT, and then there should be a treatment that involves the high dose intravenous immunoglobulin, IVIG, other anticoagulants, so no heparin anymore, and no platelet transfusion. However, if these patients are negative for both the ELISA and the platelet activation assay, then heparin treatment is possible. Now, in both cases, it's worth mentioning that because we do not know whether heparin would exacerbate the problems if these patients receive it or not. I mean, we know that in the conventional HIT, if you give heparin, then you're going to exacerbate the problem. But these patients that have not seen heparin and yet have autoantibodies circulating that are directly to platelet factor 4, if you treat them with heparin, we don't know whether these symptoms will exacerbate or not. Nonetheless, they don't want to go down that route. So they want to completely show before giving heparin treatments that these patients are negative by both these assays. Now, the British group says that, or suggests that the patients that are presented with acute thrombosis, thrombocytopenia, they need to be referred to the hematologist and then perform the prothrombin time and D-dimer level. And this is where the difference comes between both these groups, that they differentiate the affected patients by the presence of a very high D-dimer. So if the D-dimer is less than 2,000, then you have an unlikely VITT symptom. However, if the D-dimer in these patients is really high, greater than 4,000, then you are very likely, it's a high suspicion that you have vaccine-induced thrombosis and thrombocytopenia. So they actively should treat the patient with IVIG, again, avoid platelet transfusion, consider other anticoagulants, and consider treatment to increase fibrinogen level. And then you can run the HIT ELISA to confirm whether they're positive or negative. Now, if the patients are positive, then you can continue this treatment, but they are negative, you can switch to heparin treatment. So I don't know which of the strategies is more efficacious here. I kind of like the British group because they seem to be more aggressive into the treatment. But again, I don't know whether all the patients, if there is enough patients in your study that all of them consistently show high D-dimer levels, then you don't have time to run this assay. You can just proactively just treat the patient, avoid platelet transfusion, number one, and then treat IVIG so you can modulate the antibody activation and then give the other anticoagulants to stop the coagulation pathway. So I don't know, my question to you guys is, to the clinicians here, because I'm not a clinician, I'm a scientist here, what do you guys think that this is like the best strategy from the proposed groups, both the British and the German group?
Well, I think for one thing, after you have a clinical diagnosis and a pharmacological diagnosis with someone with thrombocytopenia, I think the treatment varies on the degree of thrombocytopenia. The way I would approach this, and obviously there's no hardcore data to compare the two groups, but the way I would approach it is that if there's significant drop in platelet count, then I would consider IVIG. Maybe not glucocorticoids. Steroids in medicine make everything look better. I'm not sure about that, but definitely stop heparin and heparin-containing products, including the low molecular weight heparin because of the cross-reactivity, and switch to a direct thrombin inhibitor. Now, it also depends on where the clot is. If you have a run-of-the-mill DVT, then I don't think you should go use IVIG or glucocorticoids. I think just simply stopping heparin and switching to ergotamine is sufficient. If you have pulmonary embolism, then the things change a little bit. You might have to consider plasmapheresis IVIG, specifically on submassive to massive PE. If you have a central venous thrombosis, then I also think you should consider the IVIG and the plasmapheresis, in addition to what you would do for a DVT. That's my take on it.
Mm.
I don't disagree with Erion. We both practice at the same Hartford Hospital, so it's a similar philosophy.
Yeah, I mean, most of these patients have cerebral thrombosis and splenic vein thrombosis, and some of them go into DIC. So
the DIC patient population, which is obviously marked by the hypofibrinogenemia and the elevated D-dimer and prolonged PT and bleeding time, those two, you have to throw them all you can. The German paper that you mentioned, it was heartbreaking, the case report that I presented, and it dawned on me as I was reading that paper that if this patient walked in my hospital without me being aware of these two studies that you just presented, I would have probably done the same. Someone who came with thrombocytopenia, I would have given them platelets. I would have anticoagulated. I mentioned on this side chat here that heparin is my favorite drug. I would have done the same. So a 49-year-old female that could have gone, could have been treated theoretically, was treated with quite the opposite. And, adding platelet to someone with HIT is like adding gasoline to the fire.
Yeah.
So that's another thing you should have to keep in mind.
I mean, yeah, that's why some of patients had a fatal outcome, because they were being treated with platelet transfusion and heparin.
Yeah.
And
I think it
remember,
was mostly the
this is an uncommon phenomenon. So when you're presented with it, it can be confusing, and your responses, as Erion just said, you might be tempted to do one thing which might not be the right answer.
Yeah, I mean, indeed, yeah. That's why the initial patients that were affected died, because they didn't recognize this. I mean, I don't know, how often would you see HIT, or like even suspect that a patient that received the vaccine have developed HIT symptoms? So yeah, they did the same thing. They would give an heparin and platelet transfusion, but that exacerbated the problem, and they died.
Yeah, just because, I mean, you know, everything pointed to an HIT clinical scenario. However, no one really thought that it was really truly VITT, the thing that we're talking here today.
Yep.
But one major thing that I'll take out of these two studies is that someone who's been vaccinated, and it, you know, it's always in the back of our head, and like we've been emotionally drained and exhausted by COVID, but we focus these two studies on seeing the glass half full, on treating that damn disease. So when you have a vaccine that induces an HIT, it's really, you have to have a constellation of patients' symptoms to really get into that, into that diagnosis. So it was hard initially for these patients that presented. And I think of it, when they got the vaccine, they didn't stay overnight in the hospital. So they didn't have platelets checked or a CBC checked. They just went home, and they showed up in a hospital a week later when they had petechiae and bruising. These were the favorable group. But then there were others that had portovenous thrombosis, and these were treated with platelet transfusion and heparin, which exacerbated that situation. Or there were those that had hemorrhagic stroke or central venous thrombosis. Again, these patient population, if you have a thrombosis anywhere in the human body, no one is going to say this is HIT. You know, it's just going to be, it's a knee-jerk reflex to really start them on heparin. And I think that was the issue here. And I think these two studies make me as a clinician a lot more aware of those patients that have had a COVID vaccine, especially the Johnson and the AstraZeneca, that, you know, this could be a possibility, and someone with thrombocytopenia, petechiae and whatnot. Now, I don't, you know, besides the pulmonary complication of COVID, I think in vascular surgery, we've also seen our fair share of COVID complications, such as, you know, the quote unquote term COVID feet. We've seen it, and, you know, I had to deal with one last night, but more and more we've seen this, and we've dealt with this a lot. Now we're seeing something that is induced by the vaccine that treats COVID, although in low numbers, but the outcomes could be fatal just because the late presentation of these patients.
Yeah, you know, we've had the last journal club dealing with issues regarding COVID, neurological findings now, vascular findings. We're learning about the myocarditis phenomenon that is occurring. Our knowledge will increase, but right now we just don't know that much about it. So that we can be spot on. Somebody walks in and say, oh yeah, that's it. That's going to be very difficult to do.
I have one question, if possible.
Sure.
The two vaccines are using two different types of adenovirus. I mean, is there a link between them that is being theorized? Like in terms of it's a genetically modified adenovirus. Is that correct?
Yes.
Yes.
So both are replicated defective adenoviruses.
And like, are adenoviruses that are occurring in nature? Like I saw that you mentioned prokaryotes, bacteria can cause like this PF4 activation, but are adenoviruses known to cause something like that?
So people get infected with adenoviruses, but none of them have been reported to, have HIT symptoms presented after clearing the infection.
That we know of right now.
Yes, we know that people get adenoviruses. Like I mentioned, so adenoviruses occur and they infect humans. There's human-specific subtypes, and then there's the non-human primate. So I think the AstraZeneca, the British group, used the chimpanzee adenovirus because so if we get infected with adenoviruses, then we have antibodies, right? And if we are using this as gene therapy, then we're going to lyse that adenoviral vector before it can actually do any good. So if you are using the chimpanzee, the body has never seen that chimpanzee adenovirus, so there's a likelihood of longer survival of these adenoviral particles when injected intramuscularly. Now,
Eddie, I'm sorry.
The human AD26 that's being used by J&J, that's one selective adenovirus that has a low serological agglutination across many patients that have been studied or people that have been studied. So we know that there's going to be less antibodies directed to that adenovirus itself.
Erion, could you speculate, is there any role, most of the patients in the British study, 61% were female. What's the role of progesterone, estrogen in the formation of the state?
You're asking the wrong man, but
well, have you thought about it? I don't think anybody knows.
There was one patient that had she was on oral contraceptives, and as we know, risk factors for DVT and venous thrombosis or arterial thrombosis, we've seen more and more. It's obviously obesity, smoking, and the oral contraceptives. Now, their age range wasn't, I believe, they were a relatively young population. I'm sorry?
It was 20 to 60s approximately.
Yeah. So as far as I know, and I don't want to, misspeak here, is that there's a direct correlation between OCPs and venous thrombosis. But I don't know if, endogenous estrogen, progesterone predisposes you more than a male counterpart for DVT.
Got it. Okay.
Yeah, it's still a mystery of what is really causing. I mean, like I said, in vitro studies have shown that adenoviruses can bind to platelets and activate the platelets, but that's in vitro, so it can't be completely correlated with anything we see in vivo. But at least in mice models, when adenoviruses are used for studying, they have seen some adenovirus-induced platelet activation. But again, here we can for sure say that this is the direct cause that is coming from the adenovirus. And most of their clinical trials, at least from what has been reported, they have not seen such a high incidence of VITT from the clinical trial. I mean, of course, like larger number of people that are getting vaccinated now, you might see this increase, but what is it? Is it something specific, a polymorphism, something in this genome of these patients that are making them prone to develop VITT?
So,
great question.
The one interesting fact is that they saw less and less cases with the second dose as opposed to the first one. So it seems like there's a light at the end of the tunnel, because we really don't know whether we're going to be yearly vaccinated against COVID or we're going to need a third boost or whatever. But it seems like the more you get vaccinated, the less incidence of VITT. That's something.
The likelihood that is great. Now that there's some question about the Vietnam variant that we're talking about at our hospital today, not being able, we may need a booster shot.
Yeah, that's been in the talks, yeah.
Aseda, what about Johnson & Johnson? Are there any reports of VITT?
Yes.
And
there were about 23 patients that were reported in the US. Likely none of them died.
Okay.
Probably because we started the Johnson & Johnson much later than the AstraZeneca, so they already know these symptoms that occurred in both Germany and UK as early as February.
And is it same mechanism?
Yeah, same mechanism, same thrombosis, thrombocytopenia, severe thrombosis with thrombocytopenia. I mean, the American groups have not published anything yet on their findings, whether they see these circulating autoantibodies to platelet factor 4 in these patients. But presumably it should be the same scenario, since they are presenting clinically with the same symptoms as the AstraZeneca vaccine.
So I mean, I've been getting a lot of questions from my patients as far as Johnson & Johnson vaccine about the thrombosis and everything. And of course, my answer is going to be, the risks. The benefits outweighs the risk, but I kind of wanted to know how severely is out there the thrombosis with Johnson & Johnson. And
Yeah,
I
I
couldn't
don't know
find any
that because they
study.
haven't reported any of these findings yet. So it's unclear to me. I mean, I haven't seen any publications from them.
No, me neither. I checked. Yeah.
Yeah.
May I bring up one question, or one point rather? I find it very interesting. This is such an amazing conversation, and I want to thank you for such a beautiful explanation. It just goes to show you how deep and complicated the science is, right? And when we're communicating to the patients, we did some vaccine outreach in Hartsdale at the Albanian Church here in Hartsdale, and a lot of the patients, a lot of the people in the community were, afraid of unintended consequences. And I think that the medical community very easily, especially on the lower, like we're not in the depths of the, molecular pathways, right? It's a communication problem where we kind of just, in a vague way, in a kind of way to make it as simple as possible, we eliminate a lot of validity that the community asks. When the community is saying, should I trust it, doc? Right? As simple as that. We kind of brush it aside or poo-poo it, and in one sense, we really need to take them seriously. And I think the more you're able to take them seriously, especially when we understand the pathways better, I think that allows us to have more, how can I say this, more credibility going forward. Because, there's people, and I guarantee you, that will never take this COVID vaccine. And they'll point to these events with Johnson & Johnson, with AstraZeneca to say, this is what's going on. There'll be maybe events in the future, who knows, about unintended consequences of these complicated machines that we've made, essentially. And so I just feel like we really need to be as straightforward as possible with our patients to say, hey, this is really beneficial. You're old, you have multiple medical problems. This is going to do this 100% is going to protect you. I don't know what it's going to do to you otherwise. And not to kind of break it down to like a fifth grade level, you're talking to a little child, like almost to treat them with respect and humility to say, hey, we don't know exactly 100% how things will work out, at the end.
I congratulate you for bringing the topic of getting the Albanian population vaccinated, just because they too sometimes are a little hesitant getting the vaccine for the reason you just mentioned. And I think one of the crucial points of these journal clubs, besides the fact of us banking ideas with each other and discussing a paper and enriching ourselves scientifically, we also have to tailor it into how do our background benefits from this, we have family and friends that still don't want to get the vaccine. So how do we tailor that conversation for them to really believe in this?
It's, I've worked with the Albanian Medical Federation of Europe, with the people at Yasut, and this is the crux of our problem, both here and in Albania, is reaching everyone that would trust the doctors. And the trust level for physicians in Albania is very low, sadly. But we need to be honest. I think that I didn't see who said that before, but he's right. He's spot on. Honest, clear, tell the truth, repeat it until they understand it, and not talk down to them, talk to them. I agree with you. Well said.
Just for Dr. Qaja, you asked about Johnson & Johnson and similar side effects. Belgium last week has suspended Johnson & Johnson in females below the age of 41. They had one death that they linked to Johnson & Johnson due to thrombocytopenia and blood clots recently in a female patient that was under the age of 40. So they have suspended it, and as the European Medicines Agency to further look into it and see before they can reinstitute it. This patient received it from an out in a different country and then returned back to Belgium because her employer was giving it the Johnson & Johnson vaccine. But I do agree having to be honest with patients and just admit that, yes, there are risk factors, but one of the best things is to also mention what is the risk factor of dying from COVID versus what is the risk factor of dying from the vaccine. You know, an ID doctor back in Albania and Kosovo, first when AstraZeneca came along, they were very hesitant to get it. And an ID doctor said it very well. He said, if you were to populate the entire population with AstraZeneca, you would have, based on these complications, you would have 3 or 4 people affected by these complications.
What's the denominator on that statement? 3 to 4 out of what?
Out of the entire population.
Okay.
He used Kosovo, for example, out of 2 million people.
All right.
You would have 3 or 4 people affected by it versus you have 90,000 people infected with COVID and you have over 2,000 deaths.
Just remember, 50,000 people a year die from the flu.
Of course.
We've seen a dramatic reduction in that just by washing our hands and wearing masks. So we need to keep the denominator in perspective when we tell people these things. I know that in Albania, Tritan Kalo, who runs the COVID hospital, the Infective Spital Tirana, they urge everyone to get vaccinated with whatever vaccine they can get their hands on. Because the problem is
I totally agree with that, but they also had I mean, this is specifically for Kosovo because I followed over there. They had a fluke there. They had the doctors not wanting to get AstraZeneca. And then you have a shipment of Pfizer come along, and now all the doctors are reaching for the Pfizer vaccine. So you're creating this confusion. But this is beyond this topic.
I get it, but that's neither. Yeah.
That's our culture.
That's exactly. Come on.
Culture, exactly.
Well, it's difficult to convince an Albanian that after seeing, deaths from the vaccine that, they're not going to be affected. It's like, are you going to guarantee me that if I take the vaccine, I'm not going to die? I mean,
Aseda, my cleaning lady is Albanian. She has 2 sisters that died in Albania from COVID, and she still won't take the vaccine.
oh God.
How do you convince that? How do you convince her? I've tried.
I mean, yeah, the hesitation with not only among Albanians, but I mean, elsewhere in the United States too.
Yeah.
I mean, it's interesting to me because vaccines are not the only things that produce unintended effects. Like any drug that you take can have very adverse effects. You take a PPI, you could get thrombocytopenia, So it's very interesting that vaccines just have this like very scary connotation associated with them.
They do because they have been publicized,
Yeah.
Media covers them, more people hear about them, then more fear is going to be instilled among everybody that, well, I don't want to die from a vaccine if I'm here being healthy and trying to take all my precautions so I don't get COVID-19, but I take the vaccine and I die from it. So
yeah, you're right.
It's
can I make a comment before we go too far afield? Because you're over an hour. And I said that you did a brilliant presentation. You were very clear, precise. You mentioned several times you're not a clinician, but I could not tell that listening to your presentation. You were clinically relevant. You brought all the facts. It was very clear, articulate, and well done. And I know Erion would agree with me that
I have to second that thought. I've actually I learned a lot on how to present and leave every stone unturned when it comes to a journal club just by exchanging these emails and working with Aseda. It's been a pleasure.
You lucked out, Erion. Excellent.
Aseda, you did a great job. Everybody
fantastic. Absolutely.
There's one thing before we just adjourn.
Okay.
I mean, these are the summaries. We pretty much talked about them. Again, urging to get vaccination, but there was another paper that also came from a group at UPenn in JCI, end of April, that actually tries to put complement as the culprit here for these vaccine-induced VIPT complications. And that's because complement can definitely recognize these PF4 autoantibodies. And they clearly suggest that if we can use the C3 inhibitor, which is the hallmark of the classical pathway that gets activated in the complement system, then we can, together with the IVIG treatment, also stop the overactivation of these autoantibodies. So there's another therapeutic modality here presented, which could be therapeutically important. And there are C3 complement inhibitors that have been FDA approved.
I would like to ask one question, and I want to be a little mischievous, if you allow me. But basically, here's the question: given that there is these rare complications, albeit COVID in young people, all right, we're talking about less than, let's say, 35 years old, is usually overwhelmingly not a deadly disease, even children, right? And so I guess my question is, as a community, as a medical community, given that there's, at least in the United States, the situation, we have widespread availability for seniors, for older people who are at risk, is this, knowing, like, I think this study really is like a beautiful way to understand just the complicated nature of vaccines and viruses. Is it worthwhile pushing or advocating for a vaccine in people who are younger, who may not receive much benefit whatsoever from a COVID vaccine, except the ability to travel without a lot of people looking at you, versus that risk of, God forbid, you hear a horror story of a 30-year-old, a 35-year-old who dies of some sort of clot in the brain, that horror story.
But that really has not been shown to be the case in the adolescent group, even with myocarditis, which is probably a greater risk factor, according to the CDC, for a younger under the age of 15 than this. This really has not been demonstrated in that young group. And there's a greater risk of that young person getting infected, not being sick, being a carrier, and giving it to a parent or a grandparent or someone who is not vaccinated.
Yeah, we're coming back to the same problem.
Yeah. But he said a mischievous, he was being a sheepo.
Devil's advocate.
I think, Richard, you still have a point of stating that they are at risk developing disease, the respiratory distress. Last week, I was in COVID unit. 3 out of 10 were patients up from 27 to 32 years old.
Yeah.
Risk factor, obesity. So they were all on high flow oxygen. So you still have a case stating that their life is at risk. I still believe in that lower risk, but yes, I agree with Dr. Chausha. They will be the ones who will carry the disease or the virus.
Well, I think that in this case, so definitely a proponent for vaccination. I'm just saying something different here, but something that also the German group has proposed, that because we have, the availability of the ELISAs that we can use, we can screen people, that if someone is, can have the propensity of developing this VIPT because you see these circulating autoantibodies before you get vaccination, they remove that person from receiving the AstraZeneca and give them the mRNA vaccines. So in that case, you can just really guarantee that patient that you are not going to develop these complications because of this viral vector vaccine. So, you can give some degree of confidence to these people that getting vaccinated that you might have not this complication because, or you can get the complication because this test showed this result.
Yeah, I think that would work here, but like in developed countries, but you can't really translate that to the rest of the world, unfortunately.
Why? I mean, we have, I mean, not we, but maybe America does, but
Yeah.
it should be another effort that, I don't know, we still have to extend the vaccines in Southeast Asia. Most, that seems like the higher population hasn't been vaccinated worldwide. And, you know, I don't know, maybe the cases will increase there of these VITT. And if they're limited to access to the hospitals and proper treatment for VITT, then we're going to see a lot more deaths. And here we go, the vaccines again are going to be bad again.
I don't think the risk is in getting this syndrome. I think the risk worldwide is developing variants that are not covered by the vaccine, and then we're going to see a rise in elderly people dying again.
Yeah, and also we that got vaccinated are going to get also sick again.
Right. So, you know, we have to be level-headed about this. People even died in my generation when we got polio vaccines, and polio doesn't exist anymore. But people did die when they got it.
Of course, yeah, it was very deadly, and
So-
vaccines
so-
eradicated it.
So we have to be level-headed about this. And
Yeah, I guess there's
I'm reminded by the BCG vaccine every morning, by the way.
you're right. You're right. All right, guys, this is
all right.
a great meeting. Ends
Thank
on
you so much, Aseda.
Thank you.
Aseda,
very much.
fantastic.
It was a pleasure to discuss this. Also learned a lot.
Thanks.
Well,
Thanks
before
for
we
being
end,
here.
I would like to point something. Is that okay? I just have to talk about our next journal club
Yes,
because it's
ma'am.
going to be very interesting. It's on June 16th. We have Elmira Amini. She's a general surgery resident. She will present acute mesenteric ischemia. And Clevie Goloshi, she's a first-year medical student. And the attending moderator will be Dr. Qaja again.
What do
Dr.
you know about mesenteric ischemia, Erion? You used to call us to take the bowel out. Oh, I'm going to enjoy this.
Oh, yeah.
I'm coming back, Erion, to torture you.
I think he left already.
Good.
All
All
right.
right. Well, yeah, June 16th.
We'll see you then. Thank you, Sarah.
great.
Thank you, Nora.
Take care. Bye, everyone.
Bye.
Jurgen, great job as usual. Nice to see you again.
Thanks. It's good to see you as well, Dr. Shah.
Thank you.
Jurgen is the best.
Have a good
Okay. Bye, guys.
Good night. Thank you.
Bye-bye.
Bye-bye.
How do I get out of this meeting? I always have a hard time.